Warm greeting to everyone. Today we are going to see about a very interesting topic, antibodies. They are also called as immunoglobulins. These are immunologically active serum proteins formed in response to antigen and react specifically with them. These are produced only by vertebrates. These are found in serum, body fluids and other tissues. These are synthesized only by the lymphocytes and other plasma cells. These are also called as immunological missiles. This is a basic structure of an immunoglobulin. It consists of C denotes the constant domain, consists of three constant domains and single variable domains of heavy and light chains. Then we will discuss about the types of immunoglobulins, that is the antibodies. Five major types are there. First one is the G type. It is a stable one. It consists of 4 polypeptide chained molecule formed of 2 light chains, 2 heavy chains. The light chain consists of single constant domain and a single variable domain. The heavy chain consists of 4 domains, namely a single variable domain and 3 constant domains. It is of 4 types, namely G1, G2, G3 and G4 with some structural differences. So it accounts for 70% of the antibodies. Their molecular weight is 1,50,000. Sedimentation coefficient is 7s. Half-life is 25 days. Their biological properties includes these are the only immunoglobulins that crosses the human placenta and gives passive protection to the newborns. It neutralizes the toxins and viruses, and also it acts in the absonization. Then the A type, it is found only to a very small extent in serum. There are 2 types within this A type. The first one is the serum immunoglobulin A. These are seen in secretions of respiratory, gastrointestinal and urogenital tracts. It is a monomer. It is a it has 4 polypeptide chains, 2 light chains and 2 heavy chains. Their molecular weight is 1,60,000. Sedimentation coefficient is 7s and the half-life is 6 to 8 days. Then the second type, secretory type, it is a dimer found by 2 molecules of immunoglobulin linked by a secretory component and a joining chain. This secretory component is secreted by the local epithelial cells, whereas the J chain is secreted by plasma cells. This is the structure of the secretory type of the immunoglobulin A. Here you can see the secretory component, joining chain and the other constant and variable domains. The biological properties of this type of antibody is it is an antiseptic point or mucosal point of mucous membrane. It inhibits adherence of microbe to respiratory, gastrointestinal or urogenital tracts, preventing colonizing and invading. It is present in colostrums and protects the baby from intestinal pathogens. Then the M type, these are the largest of all because their molecular weight is 9,50,000. These are pentamers, each with 4 polypeptide chains. The light chain consists of a single constant and single variable domain. Heavy chain consists of extra constant region in addition to the usual 4. Here the polymerization is present, J chain, and the sedimentation coefficient is 19 years. Carbohydrate content is 10 to 12%. Their level in serum is very low, and their half-life is 15 days, and it has a very low synthetic rate. This is a typical structure of IgM. You can see the pentamer, the 5 with each polypeptide chains. Here you can find the joining chain also. They are connected by a disulfide bond. Their biological properties includes these are the earliest antibody to be synthesized by fetus by about the 5th month. It is located in blood and protects against the blood serum infections. It plays a very important role in absonization, complement fixation, cytolysis and agglutination. Then the next type is D type. It has 2 light chains, kappa or lambda type, and 2 heavy chains, delta type. These are larger than immunoglobulin G, having a molecular weight of 1,80,000. Their half-life is 2 to 3 days. These are rich in carbohydrates. This is a typical structure of the immunoglobulin D. It consists of heavy chain, light chain of K or lambda type. Then the biological properties, it has no antibody activity, it does not mediate any effector functions, but it is associated with the surface of B lymphocytes along with immunoglobulin M. It found to act as an antigen receptor. Then the final type, which is the E type, it is E type because their heavy chain is of epsilon type. That's why these are called as E type. It consists of two light chains and heavy chains. There is no hinge here. Their molecular weight is 1,90,000. These are heat liable, whereas the rest 4 are heat stable. Their sedimentation coefficient is 8s. Their half-life is 2 to 3 days. These act in persons with allergic reactions. Their levels may increase to 50 to 100 times. Then this is the structure of immunoglobulin type E, more or less similar to the type G. Then the biological properties. These are higher level in children and helps in worm infestation and protect against the worm infestations. Then this IgE along with the antigen interaction on mast cells degranulates the mast cells and releases the vasoactive amines. The functions includes it protects against infectious diseases. The three main effector functions of all the antibodies are the opsonization, activation of complement, and the ADCC, which means the antibody dependent cell mediated cytotoxicity. We will see about these three in detail. The first one is the opsonization. Opsonization is nothing but these antibodies contain an antigen binding site and an FC receptor site. This FC receptor, what they will do means they will bind with natural killer cells and phagocytic cells. This is called opsonization. This binding starts a signal transduction pathway which results in the phagocytosis of the antigen antibody complex. Then the activation of complement. The antibodies which are involved in the activation of complement are the M and G type. Here the complement system is a system which consists of collection of serum glycoproteins with perforate membranes on activation. They perforate membranes means the antigen which are the foreign bodies, they perforate their membrane when they get activated. A byproduct of complement activation is C3b. This C3b binds non-specifically to antigen-antibody complexes. Macrophages can adhere to antigen-antibody complex, which leads to phagocytosis. So the next, the ADCC, which means the natural killer cells along with the FC receptor binded with the antibody bind with the target cells, which are the antigens, and kill the antigens. So these are the other function: agglutination, precipitating, neutralization, activations of mast cells of basophils. So herewith I have attached my references. Thank you.